Hello everyone, my name is Mia Acker. For my Evolution in the News assignment, I found popular articles on both phys.org and Science Daily about the study I'm going to tell you about today. The articles detail new information emerging about malaria's evolution. On May 21, 2018, the Nature Biology journal released a study by Thomas D. Otto et al. Regarding their research done into the evolution of Plasmodium falciparum, otherwise known as the strain of malaria that has evolved to infect humans. This study hypothesized that Plasmodium falciparum emerged around 3,000 to 4,000 years ago. The goal of this study was to find out why and how this strain of malaria emerged and how the historical evolution of the Plasmodium lineage gave rise to Plasmodium prefalciparum, which would give rise to the human-infecting strain at hand. There are several interesting findings of this study that explain Plasmodium falciparum's evolution and subsequent infection of humans. The first significant finding was that the RF1 gene complex mutated and certain genes in it were newly able to identify and bind specifically to human red blood cells. Previously, they bound to gorilla red blood cells, but the mutation made them able to only bind to humans and no longer gorillas, marking the beginning of Plasmodium falciparum's divergence and speciation. Another significant finding of the study is that Plasmodium falciparum underwent a genetic bottleneck around 5,000 years ago right before its speciation. This aligned with the agricultural revolution and therefore the arrival of more hosts to infect. Once humans began multiplying, Plasmodium falciparum had a larger host population to infect, and natural selection began to increase its effectiveness on honing Plasmodium falciparum's genes to be a more effective parasite to humans. This growing selection eventually led to its speciation between 3,000 to 4,000 years ago. The methodology of this experiment involved taking 15 milliliter blood samples from 4 gorillas and 7 chimpanzees from the park of La Lekedi in Gabon, Africa. The apes in this sanctuary were orphaned due to their parents being killed for bushmeat. Every 6 months, they are sedated for a routine health checkup. Blood samples were taken during one of the checkups and did not incur any additional stress on the animals than would have normally been endured. The levels of parasites were so low that the parasite DNA had to be isolated and replicated through PCR. The presence of multiple infections in the host posed an issue for researchers to isolate and determine separate strains of malaria in each host. The researchers used Illumina and Pacific Bioscience sequencing along with divergent evolution analysis programs to find gene variations and evolutionary patterns, also using successful bootstrapping methods. Their sequencing yielded the aforementioned results that the media went on to then publicize, that Plasmodium prefalciparum and Plasmodium falciparum diverged around 3,000 to 4,000 years ago. Originally, I was just planning on working with the Science Daily article, but after reading it and finding almost no mention of the specific findings of the study, I decided to include the phys.org article as well. The phys.org article is specifically shown here, and both articles failed to go into detail about what gene complex mutated that would lead to new speciation of Plasmodium falciparum. Specifically, the mutation was in the RF1 gene complex, and they did not discuss what its function was that led malaria to use humans as host. Specifically, that would be their identification of human red blood cells. The findings were not found to be controversial. The phys.org article briefly touched on the ethics of the blood sampling, noting that the blood was taken from orphaned primates in a Gabon sanctuary during routine checkups. The Science Daily article excluded those details and overall was the least informative article of the two. Both articles touched on Plasmodium falciparum's derived divergence of 3,000 to 4,000 years ago and the ancestral lineage's divergence at 50,000 years. That fact seemed to be the main finding that both articles found significant to specifically report on. The hooks that the articles relied on involved deadly parasite engaging language, and the energy was not equally matched in the articles, which revealed dry, vague facts about the study at hand. The implications for the future were not explored in either the study or the articles. I assume the implications are that this knowledge on lineage divergence can be used in further understanding and researching malaria in the future. The new knowledge on the genes and their specific variations among malaria species will also prove useful in future research, whether it's identifying malaria varieties among primates or tracking evolution.